There is light in the deepest parts of the ocean, not sunlight, not the glow of a research vessel's floodlights cutting through the black water, not any light that has ever touched the surface of this planet. This light was made by living things. Right now, at depths where the pressure would crush a human body in less than a second, creatures are glowing, quietly, persistently, in colors that have no name in any human language. They have been doing this for hundreds of millions of years, long before the first eye evolved to see them, long before anything on land had ever drawn a breath, and almost none of us know it is happening. Here is the number that will change how you think about light itself. Scientists estimate that between 76 and 80% of all deep-sea creatures produce their own light. In the midnight zone, the layer of ocean that begins at 1,000 meters and has never once received a single photon of sunlight, bioluminescence is not the exception. It is the rule. It is the dominant form of light on most of this planet. The ocean covers 71% of Earth's surface. Most of that ocean is deep. Most of that deep ocean is dark. And most of the creatures living in that darkness are glowing right now, at this very moment, as you listen to this. The question is not whether bioluminescence is extraordinary. The question is why it took us this long to understand that darkness was never the right word for the deep ocean at all. If you enjoy drifting into the deep ocean and discovering what lies beneath, please consider liking and subscribing. It truly helps more than you know. Are you ready? Let's begin. To understand bioluminescence, you first have to understand what it is replacing. Sunlight enters the ocean at the surface and begins to die, almost immediately. The red wavelengths disappear first within the first 10 meters. The ocean has absorbed every trace of red light. Orange follows within 20 meters. Yellow fades by 40. By 100 meters below the surface, the water around you is a deep, monochromatic blue. Not the blue of a swimming pool, not the blue of a clear sky. A deeper blue. A blue that feels like it is consuming you rather than surrounding you. At 200 meters, even that blue is gone. The last photon of sunlight, the last particle of light that has ever traveled 93 million miles from the sun, reaches no further than 200 meters into the ocean. Below that point, there has never been sunlight. Not once in the 4 billion year history of this planet. Not when the first oceans formed. Not when the first cells emerged from the primordial chemistry of ancient seas. Not ever. And yet, life did not respond to this by going dark. Life responded by making its own light. The mechanism is, in its own way, as elegant as anything biology has ever produced. Bioluminescence is a chemical reaction. An enzyme called luciferase catalyzes the oxidation of a molecule called luciferin, and the energy released by that reaction is emitted not as heat, as most chemical reactions release energy, but as light. Cold light. The most efficient form of light production that exists. An incandescent bulb converts only about 5% of the energy it uses into visible light. The rest becomes heat A bioluminescent organism converts nearly 100% of the energy in its chemical reaction into light. No heat, no waste. A living organism in the absolute darkness of the deep ocean, producing light with a thermodynamic efficiency that human engineering has never matched and may never match. This is not a curiosity. This is not a footnote in the biology of unusual creatures. This is the primary form of light production on the surface of this planet, and we discovered it, in any real sense, less than a century ago. Consider what it means for 76% of deep-sea creatures to produce light. The deep ocean is the largest living space on Earth. By volume, by surface area, by sheer three-dimensional extent, the deep ocean dwarfs every other habitat that exists. The total volume of the deep ocean, everything below 200 meters, is approximately 1 billion cubic kilometers. Compare that to all the land on Earth, including every forest, every desert, every mountain range, every square meter of habitable surface. Land covers approximately 150 million square kilometers, but only to a depth measured in meters. The deep ocean is not just larger than the land. It is larger by a factor that is difficult to state without it sounding like an exaggeration. It is not an exaggeration. In that space, that vast, cold, lightless space, the creatures are glowing. The anglerfish hangs motionless in the dark, its bioluminescent lure dangling from a modified dorsal spine above its enormous, tooth-filled jaw. The lure glows with bacteria that live inside it in a symbiotic relationship so old and so precisely calibrated that neither the fish nor the bacteria can survive without the other. The light attracts prey. Small fish and crustaceans, drawn toward what appears to be a point of light in the absolute darkness, swim directly into a trap that has been operating on the same basic principle for tens of millions of years. The anglerfish does not chase. It does not hunt in any conventional sense. It waits. It glows, and the darkness does the rest. The firefly squid Wattasenia scintillans is approximately 3 centimeters long. It has photophores, light-producing organs, covering almost its entire body. It can produce light in multiple colors. It can control each photophore independently. It can create patterns, pulses, waves of light across its tiny body with a precision that no human technology has yet been able to fully replicate or explain. When firefly squid aggregate in the bays of Japan each spring, millions of them, the sea surface turns blue. The fishermen who have watched this for generations called it the Sea of Fire. Scientists who studied it for decades struggled to understand the full complexity of the light patterns and what exactly was being communicated. There are still aspects of the firefly squid's bioluminescence that are not understood. 3 centimeters long, older than the dinosaurs, still holding secrets. The siphonophore Priadubia is the longest animal on Earth. It can reach 40 meters longer than a blue whale, longer than most ships, longer than anything that swims or walks or crawls on this planet. It is not, technically, a single animal. It is a colony of individual organisms, each specialized for a particular function, so integrated that they cannot survive independently, so coordinated that they move and behave as a single entity, and it glows. The entire length of it, 40 meters of coordinated bioluminescence, drifting in the darkness of the deep ocean, hunting and feeding with a patience and a scale that is almost impossible to hold in the mind as a single image. Most people have never heard of it. Most people will never see it. It has been alive, in some form, in these waters for an amount of time that predates almost everything we consider to be natural history. But bioluminescence is not decorative. It is not the ocean's way of being beautiful for its own sake. Every photon produced in the deep ocean costs energy. In an environment where food is scarce and every calorie is precious, no biological system survives unless it confers an advantage that outweighs its cost. Bioluminescence has survived because it works. The question of how it works, the specific functions it serves, has turned out to be far more complex than anyone initially imagined. The most intuitive function is predation. Light attracts prey in darkness. The anglerfish's lure is the canonical example, but the principle is used by dozens of different lineages of deep-sea organisms, each with their own variation on the same basic mechanism. Some use bioluminescence to illuminate prey, producing a flash of light that briefly reveals the creatures around them. Some use it as a lure, mimicking the light patterns of smaller organisms to attract predators large enough to eat. The predator becomes prey. The light that was supposed to signal safety signals danger instead. But predation is only the beginning. Counter-illumination is a strategy used by many midwater organisms, creatures that live in the twilight zone. The region between 200 and 1,000 meters, where sunlight still penetrates, faintly, as a dim downward glow. These organisms face a specific problem. Any predator looking up from below can see their silhouette against the faint light from above. They are visible from beneath, in a way they are not visible from the side or above. The solution, which evolution arrived at independently in multiple lineages, is to produce bioluminescent light from the underside of their bodies, light calibrated to match the intensity and wavelength of the ambient downwelling light. They make themselves invisible by glowing. Their bioluminescence is not a signal, it is camouflage. The complexity of this is worth sitting with. An animal, in the dim blue twilight of the ocean's middle layers, is producing light from its own body in real time, adjusting the intensity and color of that light to match the illumination coming from above, rendering itself effectively transparent to any predator looking upward. It is doing this continuously, automatically, without any conscious control. The photophores respond to ambient light levels through a system of photoreceptors and neural feedback loops that took hundreds of millions of years to develop, and that we are still, in significant ways, working to understand. Then there is communication. The deep ocean is not silent, and it is not dark, but it is the absence of most of the sensory information that land-based organisms use to navigate their world. Sound travels well in water, chemistry travels well, and light, produced internally, controlled precisely, can carry information across the distances that matter in the deep ocean. The cookie-cutter shark has a band of dark tissue on its throat surrounded by photophores. From below, in the faint, downwelling light, the dark band looks like a small fish. A larger predator, approaching from beneath, sees what appears to be a small, attractive target. When he gets close enough, the cookie-cutter shark, which is small, only about half a meter long, attacks, using its circular mouth to remove a clean, cookie-shaped plug of flesh from the much larger animal. Whales, large sharks, even submarines have been found with cookie-cutter shark bite marks. A tiny organism, using its own bioluminescence as a lure, taking bites out of creatures hundreds of times its size. The light is not incidental. The light is the strategy. Here is where the understanding of bioluminescence becomes something more than biology. For most of the history of science, the deep ocean was assumed to be biologically quiet. The pressure was too high. The cold was too extreme. The darkness was too complete. Life, scientists assumed, thinned out rapidly as depth increased, and the deepest regions of the ocean were effectively dead zones, cold, pressurized, lightless deserts where almost nothing lived. This assumption was wrong. The first deep-sea trawls in the 19th century began bringing up creatures that nobody had imagined. Strange, translucent fish with enormous eyes. Crustaceans with bodies adapted in ways that defied the logic of shallow water biology. Worms of improbable length. Jellyfish of improbable delicacy. Perfect and intact, despite having been brought up from kilometers below the surface. The ocean was not empty. But these early observations were fragmentary and difficult to interpret. The creatures brought up in nets were dead. Their bioluminescence was not visible in preserved specimens. The light they had produced in life was gone. The real revolution came when humans were able to go down. The first submersible dives into the deep ocean began in the mid-20th century, and what the scientists who made those dives encountered was not what they had expected. The darkness they had imagined, the absolute, featureless darkness of the deep, was punctuated constantly with light, flashes of blue, trails of green, pulses of white. Creatures glowing as the submersible passed, disturbed by its presence, producing light as a defensive response, as a warning, as a call to other organisms. The submersible itself, moving through the water, was triggering bioluminescent responses in everything around it. Every movement through the deep ocean produced light. Edith Witter, one of the most important marine biologists of the late 20th century, described her first dive in a submersible as the experience that changed everything she thought she understood about the ocean. She turned off the lights inside the vessel. She sat in complete darkness, pressed against the observation port, and the ocean around her was full of light. She later developed a camera system called the Eye in the Sea, a camera designed to observe deep-sea creatures without disturbing them with the artificial lights that submersibles typically use. What it recorded was extraordinary. Creatures that had never been filmed behaving naturally. Bioluminescent displays that were never triggered in the presence of submersibles because the submersible's lights suppressed them. An entire dimension of communication and behavior in the deep ocean that had been invisible because every attempt to observe it had disrupted it. This is the problem at the heart of deep-sea research. The act of observation changes what is being observed. Every submersible that has ever descended into the deep ocean has done so with lights blazing, disrupting the bioluminescent environment it was trying to study, seeing creatures in the context of artificial light rather than their own. Most of what we know about the behavior of deep-sea organisms has been gathered under conditions that are fundamentally unlike the conditions in which those organisms actually live. We have been studying them in the equivalent of a lit room when their entire lives are organized around the dark. The implications of this are still being worked through. Every study of deep-sea behavior conducted before the development of low-light and passive observation systems may need to be reinterpreted. The creatures we have been watching are not, in any meaningful sense, the creatures that exist when no one is watching. The chemistry of bioluminescence is not singular. This is one of the most remarkable facts in all of evolutionary biology. Bioluminescence has evolved independently at least 40 times. 40 separate lineages of organisms across the entire history of complex life on Earth independently arrived at the same solution, chemically producing light using different molecules, different enzymes, different metabolic pathways. The basic principle is the same. Luciferin plus luciferase plus oxygen produces light. But the specific luciferin molecules used by different organisms are chemically distinct. The luciferase enzymes are different. The control mechanisms are different. The colors produced are different. What this means, in evolutionary terms, is extraordinary. Bioluminescence is not a trait inherited from a single common ancestor and passed down through the tree of life. It is a solution that life has discovered over and over in response to the same environmental challenge. Darkness selects for the ability to produce light, and given enough time, enough generations, enough variation to select from, life produces light. Not once, not twice, at least 40 times. The diversity of bioluminescent systems reflects the diversity of the functions they serve. Blue light travels furthest in seawater. The ocean absorbs red and green wavelengths rapidly, but blue travels hundreds of meters before being significantly attenuated. Most deep-sea bioluminescence is blue, in the wavelength range of 470 to 490 nanometers, precisely the wavelength that seawater transmits most efficiently. Evolution has, over millions of years, tuned the light-producing chemistry of deep-sea organisms to the transmission properties of the medium they live in. They are not just producing light. They are producing exactly the right light for exactly this environment. Some organisms break this rule in ways that reveal the deeper complexity of what bioluminescence is doing. The dragonfish Mollucosteus niger produces red light. Red light that, in the deep ocean, should be effectively invisible. Most deep-sea creatures cannot see red wavelengths. Their eyes, adapted to the blue bioluminescence that dominates their environment, are blind to it. The dragonfish, however, can see red. It has, uniquely among known deep-sea organisms, red-sensitive photopigments. What this means is that the dragonfish has developed a private communication channel. It can produce light that only it can see. It can signal to other dragonfish or illuminate prey without that light being visible to any other organism in its environment. It has created, through the combined evolution of its light-producing system and its visual system a form of private communication in the dark. The discovery of this was made in the 1990s. It took until then because the dragonfish's red bioluminescence was so far outside what scientists expected to find that it was not initially recognized for what it was. The instruments were not looking for it. The theoretical frameworks did not predict it. The ocean, again, revealed that it operates on principles that human intuition consistently fails to anticipate. There is another function of bioluminescence that is, in some ways, the most haunting. It is called burglar alarm bioluminescence. When a small organism is attacked, when a copepod is grabbed by a fish, when a dinoflagellate is disturbed, it can produce a burst of bioluminescent light. This seems, at first, like a defense that doesn't make sense. Why produce light when you are being eaten? The light does not protect you from the predator that is already consuming you, but the light does something else. It attracts larger predators. A flash of blue light in the darkness of the ocean is a signal that something is being eaten here. And a larger predator, following that signal, may attack the thing that is eating you. The small organism, in the act of dying, produces a beacon that draws predators toward its predator. It turns the attack on itself into an attack on the thing attacking it. It is a defense that operates through sacrifice, not of the individual in any planned sense, but as an emergent property of a bioluminescent response that evolution has maintained because, across millions of individuals and millions of generations, it works. The deep ocean is organized, in part, by these flashes of light. The cascade of predation and counterpredation, the ecology of who eats whom and when, is mediated in the darkness by bioluminescence in ways that are still being mapped, still being understood, still revealing new layers of complexity with every dive and every observation. There is a quality to bioluminescence that is difficult to name. It is not the light itself. Bioluminescence, measured objectively, is faint. The glow of a single anglerfish lure, or a single dinoflagellate, or even a large siphonophore, would be invisible to human eyes, adjusted to sunlight. In the context of the surface world, it is almost nothing. A candle in a stadium, less than that. But in the absolute darkness of the deep ocean, it is everything. Every photon produced in the deep ocean is the only photon in the universe of that particular darkness. There is no background light to dilute it, no competing illumination to diminish it. When a creature glows in the deep ocean, it is creating light in a space that has never known any other kind. It is not competing with the sun. It is the only thing that has ever happened in that particular cubic meter of water. Its light is as significant in its context as any light has ever been, and most of it has never been seen. The light produced by the creatures of the deep ocean, the trillions of glowing organisms across the entirety of the world's deep water, has, throughout almost all of the history of life on Earth, being produced and consumed in absolute isolation. No eye evolved to see it except those of the creatures producing it and hunting and hiding in it. No human eye, no instrument, no record has captured most of it. It has happened, and continues to happen, in a darkness that is also simultaneously a kind of light, a light that has its own ecology, its own physics, its own 400 million year history. We have names for the things we discovered. We have measurements and classifications and theoretical frameworks. We have papers and archives and institutions dedicated to the study of bioluminescence. And still, on every dive that goes deep enough, something is seen that was not expected. Some light, in some wavelength, in some pattern, that does not fit what was already known. The ocean is generous with its secrets in the sense that it reveals them continuously. It is generous in the sense that it has so many that the revealing never ends. Right now, as this moment passes into the next, something in the deep ocean is glowing. A creature too small to see without a microscope, or something large enough to be the longest animal on Earth, or something that has not yet been named by any scientist. It is producing light in the cold and the dark and the silence, with a chemistry older than anything that has ever lived on land, for reasons that are its own. We are only beginning to understand what it means to live in that kind of dark. If this journey moved something in you, consider subscribing to Sleep on Deep. Every week, we descend a little further. The ocean is waiting. There is a place where bioluminescence becomes visible to the naked eye without any equipment, without any submersible, without descending into the deep at all. On certain nights, in certain bays around the world, the ocean surface glows blue. A swimmer moving through the water leaves a trail of blue fire. Each wave that breaks on the shore pulses with cold light. Fish moving beneath the surface are surrounded by blue halos. The effect is, for those who have never seen it, almost impossible to believe and completely natural. The organisms responsible are dinoflagellates, single-celled marine plankton, among the most abundant organisms in the ocean, producing bioluminescence as a response to mechanical disturbance. When the water is disturbed by a wave, by a swimmer, by a fish, by a boat, the dinoflagellates produce a flash of blue light. The mechanism involves a change in the electrical charge across their cell membrane, similar in principle to a nerve impulse. The disturbance triggers a cascade of chemical reactions that produces light in approximately 1/10 of a millisecond. When dinoflagellates are present in sufficient density, which they can be, in warm, nutrient-rich waters, the effect is visible with the naked eye. The bays of Puerto Rico, the shores of the Maldives, the coastline of New Zealand, the waters of the Arabian Sea during certain seasonal blooms. In these places, the ocean surface behaves as though it is producing its own moonlight, not reflecting the light of the sky, but generating light from within. This is bioluminescence operating at the scale of billions of individual cells, each one producing a single flash of blue light in response to disturbance, the aggregate of their individual responses creating something that looks, from a distance, like a continuous glow. It is not continuous. It is the sum of trillions of tiny individual events happening so rapidly and in such density that the human eye perceives them as a single sustained light. The dinoflagellates are doing this for the same reason the deep-sea organisms do it. The burglar alarm hypothesis applies here too. The flash of light attracts predators to whatever is disturbing the dinoflagellate. A fish moving through a bloom of bioluminescent dinoflagellates is effectively highlighted. Every movement it makes is marked by light. The very act of swimming through the water creates a trail that larger predators can follow. The dinoflagellates have, collectively, made darkness less safe for the things that move through them. And at the same time, they have made the ocean beautiful in a way that human beings have been noticing and struggling to describe for as long as we have been going to sea. Ancient sailors reported glowing seas and didn't know what they were looking at. Darwin, on the voyage of the Beagle, described a sea that seemed to be on fire with pale light. For centuries, the explanation was disputed electrical effects, phosphorescent salts, the decay of organic matter. It was not until the 20th century that the biological nature of the phenomenon was conclusively established. The ocean had been telling us about bioluminescence for as long as humans had been looking at it. We did not know how to hear what it was saying. 400 million years is approximately the age of the earliest known bioluminescent organisms, based on the fossil record and molecular clock analyses. This predates the dinosaurs by more than 150 million years. It predates the first forests. It predates the colonization of land by complex animal life. When the first bioluminescent organisms were glowing in the Devonian Ocean, the land surfaces of Earth were barren rock and simple vegetation, and the sea was populated by creatures whose descendants we can recognize: fish, crustaceans, mollusks, but whose world was utterly unlike any world we know. In that ocean, something discovered that light had value, that in the darkness below the surface, producing a photon was worth the metabolic cost. And that discovery, once made, proliferated. It spread not through inheritance alone, but through independent innovation, across lineages that would eventually diverge into every branch of the animal kingdom that has adapted to deep water. The evolutionary pressure that drives this is straightforward to describe and almost incomprehensible to fully grasp. Darkness does not select against sight. It selects for different kinds of sight and for different kinds of signal. A creature in the deep ocean that can produce light has, in that environment, a set of capabilities that a creature that cannot produce light simply does not have. It can lure, it can communicate, it can hide, it can warn, it can blind. The asymmetry between organisms that glow and organisms that do not is, in the deep ocean, as significant as the asymmetry between organisms with eyes and organisms without eyes on land. And this pressure has been operating without interruption for 400 million years. This means that the bioluminescent systems in the deep ocean today are not primitive. They are not simple. They are the product of 400 million years of continuous refinement, of selection and counter-selection, of arms races between predators that evolved to use light and prey that evolved to evade it, between lures and the caution that evolved in response to lures, between signals and the filters that evolved to decode or jam or ignore those signals. What we observe when we observe bioluminescence in the deep ocean is not the beginning of something. It is the current state of an evolutionary process that has been running longer than almost anything else that has ever existed. This has a consequence that is easy to state and difficult to sit with. The bioluminescent organisms of the deep ocean are, in their own domain, more optimized than we are. They have been doing what they do in the environment they inhabit for longer than anything on land has been doing anything. The anglerfish has been using its lure in fundamentally the same way for longer than mammals have existed. The siphonophore has been using coordinated colonial bioluminescence for longer than birds have existed. The dinoflagellate has been producing defensive light flashes for longer than complex plant life has covered the land. We encounter these creatures with instruments and submersibles and scientific frameworks developed over a few hundred years of systematic investigation. They encounter us with systems developed over hundreds of millions of years of survival. The encounter is not between equals. The ocean is older than our curiosity, and its inhabitants have been solving the problem of existing for longer than our entire lineage has been asking questions. There is something uncomfortable in what the history of bioluminescence research reveals about the limits of scientific observation. We sent light into the dark and called it investigation. Every submersible, every ROV, every camera system that has descended into the deep ocean has brought with it artificial illumination, because the scientists inside or behind those systems needed to see. The light was necessary, and the light changed everything it touched. The creatures of the deep ocean have evolved over hundreds of millions of years in a world where the only light is produced by living things, controlled, patterned, meaningful light that carries information about predation and communication and navigation. The arrival of a submersible with its floodlights blazing is, in the context of that world, something with no precedent. It is an event for which nothing in the deep ocean has been prepared by evolution. The responses it produces are therefore not the normal responses of these organisms to their environment. They are novel responses to a novel stimulus responses that tell us something about how these organisms react to unprecedented light sources, but not necessarily something accurate about how they live when no unprecedented light source is present. Edith Witter's work with passive observation systems began to address this problem. The eye in the sea used red light, which deep-sea organisms cannot see, for its minimal illumination needs, and observed without triggering bioluminescent responses. What it recorded in those passive observations was categorically different from what submersibles had recorded in the same depths. Behaviors that had not been observed. Densities of organisms that had not been detected. Interactions between organisms that the presence of submersibles had been disrupting or suppressing. What this suggests, and this is not a minor suggestion, is that a significant portion of what we know about deep-sea biology has been gathered under conditions that distorted what we were observing, not through incompetence or negligence, through the unavoidable fact that you cannot observe something in darkness without introducing light. And in an environment where the organisms have 400 million years of sensitivity to light as a biological signal, the act of introducing artificial light is not a neutral act. The deep ocean, in this sense, resists being known, not because it is hostile, or because the organisms in it are trying to conceal themselves from us, but because the act of knowing requires instruments, and instruments require energy. And energy in the deep ocean produces the one thing that cannot be neutral: light. Every photograph ever taken of the deep ocean has been taken in the presence of a light source that was not supposed to be there. Every documented behavior has been documented in conditions that are, from the perspective of the organisms being documented, abnormal. The natural state of the deep ocean, the thing that actually exists when no human presence is there, has never been directly observed. It has been inferred, approximated, approached. But the moment of direct observation, by definition, changes the thing being observed. This is not a problem that can be fully solved. It can be managed, minimized, worked around, but it cannot be eliminated. And sitting with this fact that the darkness of the deep ocean is, in some fundamental sense, permanently beyond our ability to fully witness, is part of understanding what bioluminescence is and what the world that produces it actually is. It is a world that has been lit in its own way for 400 million years, and we arrived recently with our own lights and called it dark. The inventory of bioluminescent organisms is not complete. It is not close to complete. Every year, deep-sea expeditions return with species that have never been cataloged. Every year, organisms previously known only from dead specimens brought up in nets are observed alive for the first time, and their behavior, including their bioluminescent behavior, turns out to be different from anything that had been inferred from dead or preserved specimens. Every year, a light is produced in some part of the deep ocean that is seen by human instruments for the first time, and the question of what produced it, and why, and what it was communicating begins the long process of being answered. The oceans contain an estimated 50,000 to 100,000 species of marine organisms that have not yet been described by science. This is an estimate with enormous uncertainty attached to it. The true number may be higher. A significant portion of those undescribed species live in deep water. A significant portion of the deep water species are likely bioluminescent. What this means, practically, is that there are creatures glowing in the deep ocean right now, whose light has never been seen by any human being, whose chemistry has never been analyzed, whose function has never been studied, whose existence has never been recorded. This is not a statement about what will eventually be discovered. It is a statement about what exists right now, in this moment, in the water that covers most of this planet. There are bioluminescent organisms alive right now that no human has ever seen. Their light is real. Their chemistry is real. Their behavior is real. And it will remain unknown until some expedition, at some unknown point in the future, happens to encounter them and happens to be equipped to observe and record them in a way that captures what they actually are. Or it will remain unknown indefinitely. The deep ocean is under no obligation to be cataloged. Its organisms did not evolve to be studied. The bioluminescence that has been operating in the darkness for 400 million years predates any observer and will, in all probability, outlast any observer. Whatever is glowing in the deep ocean tonight will continue to glow whether or not any human instrument is present to record it. The light does not require witness. It never has. There is something clarifying about this. The universe of human knowledge is large and growing, but the universe of what exists is larger, and it does not contract to fit what we have managed to observe. The deep ocean is one of the places where this asymmetry is most visible, or rather, most palpable. since visible is not quite the right word for a place defined by the absence of the kind of light that makes sight possible. What bioluminescence tells us, in the end, is that darkness is not the absence of life. It is not the absence of activity or complexity or communication or beauty. It is the absence of sunlight, and life found other light. Life produced its own. Life has been producing its own light in the darkness that sunlight cannot reach for longer than the ancestors of every land-dwelling animal have existed. The deep ocean is not waiting to be explored. It is not incomplete, not unfinished, not a darkness that needs to be filled with human observation to become real. It is complete. It is lit in its own way. It has been for 400 million years. We are the ones who are still learning to see. The ocean is not uniform. It is layered not just by temperature and pressure and salinity, but by light and by the organisms that have adapted to each layer's specific luminous conditions. The sunlit zone, the epipelagic layer from the surface to approximately 200 meters, receives enough sunlight for photosynthesis. This is the layer most people think of when they think of the ocean. It is the layer where coral reefs grow, where the largest fish school, where the blue whale feeds. Bioluminescence exists here, but it is dominated by the dinoflagellates organisms that glow in response to disturbance, producing the surface glows that sailors have been observing for millennia. The function of bioluminescence in the sunlit zone is different from its function below, because the context is different. Sunlight is the dominant energy source. Light is abundant. Bioluminescence is one signal among many. Below this, from 200 to 1,000 meters, is the twilight zone, the mesopelagic layer. Here, sunlight penetrates as a faint, diffuse, downwelling glow, shifting toward blue as depth increases, fading. Animals at the upper boundary of this zone live in dim blue light. Animals at the lower boundary live in near total darkness. The organisms of the twilight zone have evolved in the context of this gradient, with eyes capable of detecting very low light levels, with counter-shading and counter-illumination to manage their own visibility in the ambient downwelling light, with bioluminescent systems calibrated to function in an environment that is neither fully dark nor meaningfully bright. This is the most densely populated layer of the deep ocean. It contains the largest fish populations below the sunlit zone. It is also the zone that performs one of the ocean's most important ecological functions, the biological pump. Every day, enormous quantities of organic material, dead organisms, fecal pellets, shed tissue sink from the sunlit zone into the depths below. This is called marine snow. It is the primary mechanism by which carbon fixed by photosynthesis at the surface is transported into the deep ocean, where it can be sequestered for centuries or millennia before returning to the surface. The biological pump is one of the primary mechanisms by which the ocean moderates the concentration of carbon dioxide in the atmosphere. Without it, the planet would be significantly warmer. The organisms of the twilight zone are a critical part of this pump. They feed at or near the surface at night, consuming phytoplankton and other surface organisms, then descend to the twilight zone during the day. When they die, or when they are eaten and their predators die, Their bodies carry the carbon they contain down into the deep carbon that was at the surface, now sequestered in the dark. The daily vertical migration of these organisms, billions of animals moving up and down by hundreds of meters every 24 hours, is one of the largest animal migrations on Earth, and many of these organisms are bioluminescent. The relationship between their bioluminescence and their role in the biological pump is not fully understood. It is not clear, for instance, whether bioluminescent signals play a role in coordinating the vertical migrations, or whether they are used for communication during the migration itself. It is not clear whether the predation dynamics in the twilight zone, which are mediated in part by bioluminescence, affect the efficiency of the biological pump. These are open questions. They are important questions. The biological pump is a process that matters for the entire planet, and we do not fully understand how bioluminescence fits into it. Below the twilight zone, below 1,000 meters, is the midnight zone, the bathypelagic layer. Here, there is no sunlight at all, not even a trace. The only light is bioluminescent. And it is here that bioluminescence achieves its most sophisticated forms. The organisms of the midnight zone have been cut off from sunlight for the entirety of their evolutionary history. There is no selection pressure here for eyes that can detect sunlight, because there is no sunlight to detect. The eyes that exist here are optimized for bioluminescent wavelengths. The photopigments that fill them are sensitive to blue light in the range that the ocean transmits most efficiently. Some organisms have enormous eyes relative to their body size, and eyes that can collect the faint bioluminescent light from considerable distances. Others have essentially abandoned vision in favor of mechanosensory systems detecting the pressure waves produced by movement rather than light. Still others have eyes positioned upward, watching for the bioluminescent signals of prey above them. The ecology of the midnight zone is organized around bioluminescence in a way that has no parallel in any other ecosystem on Earth. Light, in this context, is not background, it is signal. Every photon is produced deliberately or semi-deliberately by a living organism. Every flash has a sender. Every pulse has a context. The darkness is not featureless. It is full of information, structured by bioluminescent communication that has been developing for longer than any land-based ecosystem has existed. Not all bioluminescent light is produced by the organism that carries it. The anglerfish's lure contains bacteria. This is, on its face, a simple statement. It becomes extraordinary when you consider what it means. The anglerfish cannot produce bioluminescent light on its own. It has no internal chemistry capable of generating photons. What it has is an organ, the esca, the bulbous lure at the tip of its modified dorsal spine, that is hollow, and that is filled with bacteria, specifically with Alivibrio fischeri and related species. These bacteria produce bioluminescent light. The anglerfish provides the bacteria with shelter and nutrients. The bacteria provide the anglerfish with the light that makes its lure function. Neither can do what they do together without the other. This is symbiosis. But the depth of the relationship goes beyond what the word typically suggests. The anglerfish's lure is not simply a container for bacteria. It is a biological optical system. The structure of the esca includes reflective tissues that direct and concentrate the bacterial light. It includes a lens-like structure that focuses the light into a directed beam. It includes tissues that can modulate the intensity of the light not by controlling the bacteria directly, but by controlling blood flow to the esca, which in turn affects the oxygen supply to the bacteria, which affects their light production. The anglerfish has built around a colony of bacteria a controlled optical instrument of remarkable sophistication. And the bacteria, for their part, are not passive. They are among the best studied of all bioluminescent organisms, and their light production is regulated by a mechanism called quorum sensing. Individual Vibrio fischeri bacteria produce very little light, but when they are present in sufficient density, when there are enough of them, their light production increases dramatically. They detect the density of their own population by releasing and detecting chemical signals. When the concentration of those signals exceeds a threshold, the entire population switches from low light to high light production simultaneously. The light of the anglerfish's lure is, in this sense, the collective decision of a bacterial community, made through a form of chemical democracy. Quorum sensing, since its discovery in bioluminescent marine bacteria, has been found to regulate a wide range of bacterial behaviors beyond light production. The implications for understanding bacterial ecology and for medicine, since quorum sensing is involved in the formation of bacterial biofilms and in virulence, have been significant. A discovery about the mechanism of bioluminescence in deep-sea anglerfish contributed to fundamental understanding of how bacteria communicate and coordinate their behavior. The deep ocean is not an isolated system. It is connected in ways that are still being mapped to knowledge about biology that applies far beyond it. There are other symbiotic, bioluminescent relationships. Some squid carry Vibrio fischeri in specialized light organs and use the bacterial light for counter-illumination, producing light from their underside that matches the downwelling ambient light, rendering them invisible to predators below. The Hawaiian bobtail squid has been studied extensively and has become one of the model organisms for understanding symbiosis in general. It hatches without bacteria, acquires them from the surrounding seawater within hours of birth, and spends the rest of its life managing a colony of light-producing microorganisms in an organ designed for exactly this purpose. The ocean, in this way, is not just a collection of individual organisms performing individual functions. It is a system of relationships, some of them so old and so integrated that the distinction between organism and symbiont has become nearly meaningless. The anglerfish and its bacteria are not two organisms that happen to share space. They are a single functional unit, the product of an evolutionary relationship older than most of what we consider to be complex life. Bioluminescence did not emerge from individual organisms deciding, one by one, to produce light. It emerged from a world in which organisms are never truly individual, in which the boundaries between organisms are permeable and the line between self and other has always been drawn differently by biology than it is drawn by human intuition. The light in the deep ocean is not produced by lonely creatures glowing in isolation. It is produced by communities, by relationships, by systems that have been refining themselves for 400 million years in the cold and the dark and the silence that surrounds us right now, beneath the ocean that covers most of this world. What does it mean to produce light in a place where no light has ever come from outside? The bioluminescent organisms of the deep ocean are, in one sense, creating something unprecedented in their environment with every photon they produce. The deep ocean, before life produced light in it, was a place where no photon had ever existed. There was no light. There was pressure and cold and chemistry and the slow drift of organic material down from above, but no light. And then, at some point in the Cambrian, or slightly before, a chemical reaction that released energy as photons rather than as heat persisted in a population of organisms because it turned out, in that environment, to be useful. And light entered a world that had never had it. Not sunlight filtered down from above. Light that was created there, in the dark, by organisms that found that producing photons was worth the metabolic investment. This has a parallel, at a vastly different scale, with the emergence of photosynthesis. Before photosynthesis, the surface of the earth received sunlight, but life could not use it directly. When cyanobacteria developed the ability to convert light energy into chemical energy approximately 3 billion years ago, they transformed the surface of the planet. The oxygen they produced as a byproduct accumulated in the atmosphere, creating the conditions that make complex aerobic life possible. Photosynthesis brought light into the chemistry of life at the surface. Bioluminescence brought life's chemistry into the production of light in the deep. These are not the same event, but they share something: the moment at which light and life entered a new relationship. The moment at which a photon became meaningful, not just as energy falling from above, but as something that living things produced and controlled and used. The deep ocean is not a place that is waiting for light to arrive. It has its own light. It has had its own light for longer than complex life has existed on land. The darkness that we observe from the surface, and that seemed to early biologists to make the deep ocean inhospitable, is not the darkness of absence. It is the darkness of a different kind of illumination. A bioluminescent world, organized around different wavelengths, different intensities, different functions than anything that the surface has produced. We are, as a species, attuned to sunlight. Our eyes evolved in it. Our sense of beauty evolved in it. Our concept of day and night, of visibility and concealment, of bright and dark, is organized around the rhythm of the sun. The deep ocean operates on no such rhythm. There is no day there, no night, no season in any sense that the surface knows. The light in the deep ocean is not cyclical. It is incidental produced when organisms produce it, for reasons of predation and communication and defense, and absent when they do not. To imagine what it is like to live in that world, not as a tourist, not as a visiting scientist in a submersible, but as an organism for whom bioluminescence is the primary perceptual reality, is to imagine a relationship to light that is almost the opposite of our own. We are surrounded by light and seek darkness for rest. They are surrounded by darkness and produce light when they need to navigate it. There is no darkness in their world, because darkness is a concept that requires a prior experience of light to have meaning. For them, there is only the dark that is always present, and the light that they make, briefly, when it serves them, and the signals of other creatures moving through the same absence. The deep ocean is not a silent void. It is a conversation conducted in photons between organisms that have been refining their language for 400 million years. We have been listening for about 60. And still, on every dive, something new is said. The history of how humans came to understand bioluminescence is, in some ways, a history of how long it takes to accept that nature operates by rules we have not yet imagined. The earliest written records of glowing seas appear in ancient Greek texts. Aristotle, in the 4th century BCE, noted that dead fish and flesh sometimes appeared to glow in the dark, and that certain jellyfish produced light. He had no explanation. The phenomenon was catalogued and set aside. For the next 2,000 years, it remained in the category of curiosities, things that happened, that were observed, that could not be fully explained. The explanation that persisted longest was phosphorescence, a term that implied a connection to phosphorus, the element which does in fact glow when oxidized in air. Glowing seas were called phosphorescent seas. The phenomenon was attributed to phosphorus released from decaying organic matter, or to electrical effects in the water, or to undiscovered minerals. The biological origin of the light was recognized in some contexts. Jellyfish were clearly alive and clearly glowing, but the connection between biological activity and the broader phenomenon of glowing seas was not consistently made. Charles Darwin, on the HMS Beagle in the 1830s, encountered bioluminescent seas in the South Atlantic and described them in terms that combined scientific observation with a kind of awe that is difficult to suppress even in retrospect. The ship was moving through water that appeared to be burning with pale fire. The bow wave glowed. The wake stretched back into the darkness as a line of cold blue light. Every dip of the oars produced a flash. Darwin noted the biological nature of the phenomenon. He could see individual glowing points in the water, organisms small enough to be microbes, but the full understanding of what he was seeing was not available to him. The systematic study of bioluminescence as a biological phenomenon began in earnest in the 19th century, with the work of Raphael Dubois, a French pharmacologist who worked in the 1880s and 1890s. Dubois ground up bioluminescent clams, Pholus dactylus, a marine mollusk that glows when disturbed, and separated the crude extract into two fractions by alternating exposure to hot and cold water. He found that neither fraction alone could produce light, but that combining them produced a glow. He named the heat-stable fraction luciferin, from the Latin for light-bearer, and the heat-labile fraction luciferase. He had, without fully understanding it, identified the two components of the bioluminescent reaction, the substrate and the enzyme. This was 1887. The mechanism of life's ability to produce light was identified in crude outline before the electron was discovered, before DNA was understood, before the structure of proteins was known. Dubois had the right answer about the basic chemistry, without any of the framework that would later be necessary to understand what luciferin and luciferase actually were at a molecular level. The full elucidation of the bioluminescent reaction took another 80 years. Newton Harvey, an American biologist who spent most of his career at Princeton, dedicated decades to the systematic study of bioluminescence across different organisms, cataloging the variety of light-producing systems and establishing the chemical principles common to them. His book, Bioluminescence, published in 1952, was the first comprehensive treatment of the subject and remains a foundational reference. Harvey's work established something important, that bioluminescence was not a single phenomenon, but a family of related phenomena, sharing the basic principle of luciferin-luciferase chemistry, but varying enormously in the specific molecules involved. The luciferin used by the firefly is not the same molecule as the luciferin used by dinoflagellates. The luciferase of a deep-sea fish is not the same enzyme as the luciferase of a jellyfish. The word luciferin is not the name of a specific molecule. It is the name of a class of molecules substrates that can be oxidized by their corresponding luciferases to produce light. This matters because it means that bioluminescence has not been inherited from a single common ancestor. It has been invented independently, many times, by organisms that happen to have precursor molecules that could, with sufficient modification, serve as substrates for light-producing reactions. The evolutionary pathways to bioluminescence are as diverse as the organisms that produce it. The implications of this for our understanding of evolution are significant. Evolution does not always converge on the same solution to the same problem by the same route. Sometimes it converges on the same solution by entirely different routes, arriving at the same destination through different chemistry, different genes, different developmental pathways. Bioluminescence is one of the clearest examples of this convergent evolution at a biochemical level, and studying it has contributed to a broader understanding of how evolution can produce similar outcomes in unrelated lineages. The science of bioluminescence did not stay in the ocean. In 1962, Osamu Shimomura, a Japanese-American chemist working at Princeton University's Marine Biological Laboratory, extracted a protein from the jellyfish Equirria victoria that fluoresced green when exposed to ultraviolet light. He called it green fluorescent protein, or GFP. He spent years studying it without any clear idea of what it would eventually be used for. He was interested in it because it was interesting. The jellyfish glowed. He wanted to know how. The protein he extracted is not, strictly speaking, bioluminescent in itself. It is fluorescent. It absorbs light of one wavelength and emits light of a longer wavelength. In the jellyfish, it works in conjunction with a bioluminescent protein called acrine, absorbing the blue light that acrine produces and converting it to the green light that the jellyfish emits. But the fluorescent protein itself turned out to be something remarkable, a genetically encodable light source. It was a protein that, when the gene encoding it was introduced into another organism, would cause that organism to glow green. Martin Chalfie, a biologist at Columbia University recognized the potential of this in the early 1990s. He inserted the GFP gene into the nematode Caenorhabditis elegans, causing specific cells in the transparent worm to glow green under ultraviolet light. For the first time, it was possible to watch specific cells in a living organism to mark them with light and observe them as they moved and divided and differentiated. This was a revolution in cell biology. It made visible things that had been invisible. It made the dynamics of cells and living tissue directly observable in a way that no previous technique had allowed. Roger Cian, a chemist at the University of California, San Diego, subsequently developed variants of GFP that fluoresced in different colors: cyan, yellow, red, orange. By using multiple variants in the same experiment, it became possible to label multiple different cell types simultaneously, each glowing in a different color, allowing their interactions to be observed directly. The palette of fluorescent proteins that Cian developed transformed not just cell biology, but neuroscience, developmental biology, immunology, and cancer research. Every field that needed to track specific cells in living tissue was transformed by the ability to mark those cells with light. Shimomura, Chalfie, and Cian shared the 2008 Nobel Prize in Chemistry for the discovery and development of GFP. The discovery that led to the Nobel Prize began in a jellyfish. The jellyfish was bioluminescent. The bioluminescence was being studied because it was interesting in a remote marine laboratory by a scientist who had no particular expectation that what he was finding would change the practice of cell biology. This is the way that basic scientific research frequently works, and it is the reason that basic research conducted out of curiosity rather than toward a predetermined application remains essential. The value of understanding bioluminescence could not have been predicted from the study of bioluminescence itself. It became visible only when the knowledge was in hand and someone recognized its potential. There are other applications. Bioluminescent reporter genes, genes that cause cells to glow when they are active, are used throughout biology to study when and where genes are expressed. Bioluminescent bacteria are used to detect environmental contamination. Some strains glow more brightly in the presence of specific pollutants, making them effective sensors for monitoring water quality. Bioluminescent organisms are being studied as potential sources of light in applications where conventional lighting would be problematic. For instance, in environments where electrical sparks would be dangerous, or in situations where biological lighting might be more sustainable than electrical lighting. The chemistry of life's ability to produce light is now a tool, a set of tools, deployed across dozens of fields, in applications that range from fundamental biological research to medical diagnostics to environmental monitoring. All of it traces back to organisms in the ocean, producing light in the dark, for reasons that had nothing to do with human applications and everything to do with survival. The ocean has been doing chemistry for 4 billion years. We have been watching for a fraction of that time, and what we have found by watching has already transformed what we can do. The question of what we have not yet found, what chemistry is still operating in the dark in the organisms we have not yet studied, in the reactions we have not yet catalogued, is one of the more genuinely open questions in all of science. There is a question that bioluminescence raises that is not, strictly speaking, a scientific question. It is a question about meaning and about the relationship between observation and existence. The light produced by a dinoflagellate, or an anglerfish, or a siphonophore in the deep ocean serves functions. It attracts prey, or communicates with mates, or defends against predators, or provides camouflage. In this sense, it has meaning. It produces effects in the world. But it has this meaning whether or not anyone is watching. The anglerfish's lure has been attracting prey for tens of millions of years without any observer present to register what was happening. The meaning was there before the observation. This is different from the way we often think about beauty or significance as qualities that require a perceiving mind to exist. We tend to think that a sunset is beautiful because someone is watching it. We tend to think that a piece of music is meaningful because someone is listening. But the bioluminescence of the deep ocean suggests a different relationship between significance and perception. The light is meaningful. It is functionally meaningful, ecologically meaningful, evolutionarily meaningful, regardless of whether any human mind is present to appreciate it. In this sense, bioluminescence is a small piece of evidence for something that science generally assumes, but that is philosophically complex. The world exists and operates by principles that have meaning, independent of observation. The deep ocean does not become less organized or less complex or less illuminated in any important sense because no human is there to observe it. The patterns are there. The chemistry is there. The light is there. We find this easy to accept as an abstract proposition. We find it harder to fully integrate emotionally. The human mind is, by its nature, observer-centric. We experience the world from inside a perspective, and it is difficult to genuinely imagine the world as it exists without any perspective at all. But the deep ocean, with its bioluminescent light that has been operating for 400 million years without any observer, is a persistent reminder that the world is not organized around our perception of it. It was here before us. It operates according to its own principles. And when we observe it, we are not conferring meaning on it. We are encountering meaning that was already there. There is something restful about this if you sit with it long enough. The pressure that human consciousness sometimes feels the sense that it must be present, must be attentive, must be observing, or the world will somehow lose its significance, is not well founded. The deep ocean has been significant for 400 million years without us. It will continue to be significant. The anglerfish's lure will continue to glow. The siphonophore will continue to drift. The dinoflagellates will continue to flash in the waves. Not for us, not despite us, independent of us, in the way that everything that has existed for hundreds of millions of years is ultimately independent of the creatures that arrived most recently to observe it. This is the gift that the deep ocean offers, if you let it. Not the comfort of being watched over or protected or understood. The comfort of being temporary in a world that is not. The comfort of existing in something vast and old and illuminated in ways that our eyes are only beginning to learn to see. The deep ocean does not need us to appreciate it. It is complete without us. And in that completeness, there is something that feels, in the darkness and the silence, very much like rest. The organisms that have contributed most to our understanding of bioluminescence are not the most spectacular ones. They are the most convenient ones. Illuminate fishery, the bacterium that lives in the light organs of the Hawaiian bobtail squid, and the anglerfish's lure, has been studied more intensively than almost any other bioluminescent organism. It grows easily in laboratory conditions. It can be cultured in large quantities. Its genetics are tractable. Its quorum sensing system, discovered in part through the study of its bioluminescence, has been dissected at a molecular level in extraordinary detail. We know more about how Vibrio fishery produces light than we know about how most of the visible ocean produces light, simply because it is convenient. The firefly, a terrestrial bioluminescent organism, not a marine one, has been studied intensively for similar reasons of convenience. It is large enough to handle. Its light organ is accessible. Its bioluminescent reaction can be reproduced in cell-free systems. The luciferase gene from the firefly, Photinus pyralis, was among the first reporter genes used in cell biology, and it remains one of the most widely used tools in molecular biology. Millions of experiments conducted every year in laboratories around the world use firefly luciferase as a reporter. The firefly, a small insect that uses light to attract mates in the summer evenings of the northern hemisphere, has become one of the most important model organisms in biochemistry. The Eichoria victoria jellyfish, source of GFP, was convenient because it is abundant in Puget Sound during certain seasons, and the Friday Harbor laboratories on San Juan Island provided a base from which Shimomura could collect large numbers of them. The discovery of GFP was dependent not just on Shimomura's skill and curiosity, but on the geographical and logistical accident that a bioluminescent jellyfish happened to be abundant near a well-equipped research station. The deep-sea organisms that produce the most sophisticated and varied bioluminescence, the dragonfish, the siphonophores, the midwater crustaceans, the transparent, gelatinous organisms of the midnight zone are almost entirely intractable. They cannot be maintained alive in laboratory conditions. They die during collection. Their light organs degrade rapidly after death. The chemistry of their bioluminescence can only be studied from samples collected during rare deep-sea expeditions, and the behavioral context of their bioluminescence, what the light is actually doing in their lives, can only be observed in situ, in the deep ocean, under conditions that are technically demanding and enormously expensive. The consequence is that the organisms we understand best are not the most ecologically important ones. The organisms that dominate the bioluminescent ecology of the deep ocean that produce the bulk of the light in the largest living space on Earth are precisely the ones that are most difficult to study. We have a detailed understanding of the molecular biology of firefly luciferase and a fragmentary understanding of the bioluminescent chemistry of most deep-sea organisms. We have a thorough understanding of quorum sensing in Aliivi Brio fishery and a poor understanding of the sensory systems that detect bioluminescent signals in the midnight zone. This is not a failure of effort. It is a structural feature of the relationship between scientific investigation and the ocean. The ocean does not present its most complex phenomena in the most accessible locations. The most interesting biology, from the perspective of evolutionary complexity and ecological significance, is the biology that is hardest to reach. And the technology required to reach it and to observe it without disrupting it is still, in many respects, being developed. There are deep-sea biologists who have spent their entire careers working toward a single type of observation that has not yet been achieved. There are research programs designed around the development of instruments that will allow passive observation of bioluminescent behavior in the midnight zone at the temporal and spatial resolution needed to actually understand what is being communicated. These programs take years. The instruments fail. The dives are canceled. The specimens are lost on the way up. The funding runs out. And then, sometimes, something works. A new camera system captures a behavior that has never been seen. A new analysis technique reveals a pattern in existing data that was not recognized. A new expedition brings back a specimen intact enough to study. The science of bioluminescence is a science of patience. Not the patience of waiting for something to happen, but the patience of working steadily and carefully and with frequent failure toward an understanding that reveals itself incrementally through the accumulation of many partial observations over decades. This is what serious science looks like. Not the dramatic moment of discovery, though those happen, but the long accumulation of work that makes the dramatic moments possible. The work done at sea, in difficult conditions, by people who have chosen to spend their professional lives trying to understand a phenomenon that occurs in one of the most inaccessible environments on Earth. In the autumn of 1997, a research team from the Monterey Bay Aquarium Research Institute was conducting a routine ROV dive in Monterey Canyon off the coast of California. The ROV's cameras were running. The depth was approximately 900 meters on the lower boundary of the twilight zone, where the last traces of sunlight disappear. The ROV disturbed something in the water. What happened next was recorded on video and has been studied for the decades since. A cloud of bioluminescent material expanded outward from the disturbance, not the small flash of an individual organism, but a diffuse, expanding sphere of blue light that hung in the water for several seconds before fading. The researchers were uncertain what had produced it. The resolution of the footage was not sufficient to identify the organism or organisms responsible. The event was logged and filed. Years later, re-examination of the footage in the context of improved understanding of mid-water bioluminescence led to the tentative identification of the light cloud as a defensive response by a siphonophore or a colonial organism of similar structure, a bioluminescent display, large enough and sustained enough to potentially startle or deter a predator significantly larger than the organism producing it. What makes this story interesting is not the observation itself. It is the process of understanding. The event was observed in 1997. Its possible interpretation was not developed until years later, when enough had been learned about the organisms present at that depth and about the physical properties of the light cloud to make a tentative identification plausible. The observation preceded the understanding by years. The ocean had produced something, and it took time to develop enough context to begin to understand what had been produced. This is the characteristic experience of deep-sea research. The observation happens first. The understanding accumulates slowly through the collection of many other observations, the development of better instruments, the accumulation of ecological and behavioral context. The ocean reveals, but does not explain. The explanation is something that researchers construct over time from the fragments that expeditions provide. There is a word in Japanese ma that refers to the meaningful space between things, the pause between notes in music, the empty space in a painting that gives the occupied space its significance, the interval between question and answer that allows meaning to develop. The concept resists direct translation, but describes something that is recognizable once you know to look for it. The deep ocean has something like ma. The spaces between the lights, the darkness between the flashes of bioluminescence are not empty. They are the context in which the lights are meaningful. A flash of blue in absolute darkness is visible precisely because of the darkness that surrounds it. The darkness is not absence. It is the medium in which the signal exists, the background without which the signal cannot be distinguished, the silence that gives the light its significance. When we think of bioluminescence, we tend to think of the light. We catalog it, measure it, study its chemistry and its functions. But the darkness that makes bioluminescence what it is, the 400-million-year-old darkness of the deep ocean, the darkness from which no sunlight has ever been absent because sunlight has never been present, is as much a part of bioluminescence as the light itself. You cannot understand one without the other, and you cannot understand either without sitting with them together for long enough that the boundary between them becomes something other than a simple contrast. The deep ocean is dark. The deep ocean glows. These are not contradictions. They are the same fact, seen from different distances, at different moments, through different instruments. The organisms that live there have always known this. They have organized their entire existence around it. For 400 million years, they have been moving through a world where darkness and light are not opposites but partners, where the darkness is what makes the light meaningful, and the light is what makes the darkness navigable. We are learning this now, slowly, in increments, through instruments that disturb what they observe, from a distance that is always too great for full understanding, with a patience that is still developing. But we are learning. And as you drift towards sleep, as the darkness that surrounds you deepens, and your awareness narrows to something quiet and undirected, know that somewhere below the deepest ocean surface, in water so cold and dark and pressurized that no human body could survive it for a moment, something is glowing. It has been glowing since before your ancestors walked on land. It will be glowing after every institution and language and memory you have ever known has been dissolved back into the chemistry of the world. The light in the dark ocean is not waiting for us. It is not for us. But in the way that the most ancient things sometimes are, it is if we are quiet enough and still enough and patient enough to receive it deeply and inexplicably reassuring. The ocean is not empty. It never was. If you could map every bioluminescent event in the ocean simultaneously, every flash, every glow, every pulse produced by every organism at every depth, at a single moment, the resulting image will look nothing like what we typically imagine when we picture the ocean. The surface would be speckled with blue dinoflagellates, responding to the movement of waves and fish and wind, producing their brief defensive flashes in the shallow waters of warm seas. Around coral reefs, the pattern would be complex. Organisms in the reef structure producing light that interacts with the light produced by organisms in the water column above them, creating a layered, luminous ecology that has barely been studied. In the open ocean surface waters, the pattern would be sparse individual organisms separated by meters or tens of meters of dark water, their flashes isolated, responding to specific stimuli. Deeper, in the twilight zone, the pattern would change. Here, the organisms are more diverse and their bioluminescence more varied. Counter-illuminating organisms producing a diffuse, downward glow that matches the ambient light. Midwater fish flashing warning signals. Siphonophores producing long pulses of blue as they drift and hunt. The density of bioluminescent events in the twilight zone per cubic meter of water would be higher than at the surface because the organisms here are more dependent on light they produce themselves, and because the competition for resources and the pressure from predators are intense. Below 1,000 meters in the midnight zone, the pattern would be different again. Here, the bioluminescence is not ambient background. It is punctuated, deliberate, specific. The anglerfish's lure glows continuously, but faintly, a persistent light source in the dark, moving slowly, attracting and waiting. When prey approaches, the light continues. The trap operates on patience. Around the anglerfish, at distances measured in tens of meters, other organisms respond to the light attracted, repelled, or simply oriented by it in the featureless dark. Further away, a dragonfish is hunting with its red bioluminescence, red light that is invisible to every other organism in its environment, a private spotlight that illuminates its prey while remaining undetectable. The prey continues to behave normally, unaware of the illumination. The dragonfish approaches. The encounter is, from the prey's perspective, from the dark. At the same moment, at a depth of 2,000 meters, a siphonophore 40 meters long is drifting. Its bioluminescent tentacles trail behind it in the current. When a fish or crustacean contacts a tentacle, the bioluminescence intensifies a bright pulse of blue that moves along the length of the organism toward its feeding polyps. The organism is signaling to itself, coordinating the activity of its colonial members through bioluminescence, in the same way that a nervous system coordinates the activity of individual cells through electrical signals. And at the very deepest levels at the hadal depths, below 6,000 meters, in the trenches where the pressure exceeds 600 atmospheres and the temperature is barely above freezing even here, life exists. And even here, some of it glows. The specific bioluminescent organisms of the hadal zone are among the least studied in all of marine biology. The expeditions that have reached these depths have been few. The time spent at these depths has been brief. The observations have been fragmentary. But the organisms are there. The chemistry is there. The light is there, in the deepest places on Earth, where no instrument has yet fully mapped it and no comprehensive study has yet been done. The full bioluminescent map of the ocean has never been made. It cannot currently be made. It would require instruments deployed simultaneously across all depths, in all locations, observing without disturbing, recording without light pollution. The technology does not exist. The logistics are impossible. The map remains theoretical. A thing we know would be extraordinary if we could see it, but that we have never and may never see in its entirety. What we have instead are fragments, ROV observations in specific locations, at specific depths, trawl samples that preserve chemistry but not behavior, acoustic data that reveals the presence of organisms but not their light, satellite observations that can detect surface bioluminescence from orbit in some conditions, and the testimony of scientists who have been in submersibles at depth, looking out through small portholes at a world that was doing something they had not expected, trying to record and describe what they were seeing in real time. These fragments, accumulated over decades, constitute what we know. They are extraordinary fragments. They contain enough information to have transformed multiple fields of biology and chemistry. They have revealed organisms and mechanisms and behaviors that were not imagined before they were observed. And they are a tiny fraction of what is there. There is a moment, familiar to anyone who has ever lain awake in a completely dark room, when the darkness becomes something other than the absence of light. When your eyes, having found nothing to fix on, begin to produce their own input, the faint phosphine patterns of the visual cortex working without stimulus, the mild hallucinations that appear in the peripheral vision when there is nothing to anchor it. In that moment, the darkness is not nothing. It is something, a space that your nervous system, evolved for a lit world, finds uneasy and begins to fill with its own productions. The darkness of a room at night is, neurologically, an experience. It has texture and depth that are not actually there. It produces responses in the brain that are designed for a world where darkness meant danger and where the night was full of threats that human senses were not well equipped to detect. The organisms of the deep ocean have no such response to darkness. Darkness is their native medium. Their nervous systems, their sensory organs, their behavioral patterns are not designed for a lit world in which darkness is a temporary and potentially dangerous absence of normal conditions. They are designed for a world in which light is a rare, specific, meaningful event produced by living things for specific purposes in the middle of a medium that does not otherwise contain it. When an anglerfish hangs in the dark with its lure glowing, it is not experiencing anything we would recognize as waiting in the dark. It is simply existing in its medium, doing what it has evolved to do. There's no contrast for it between light and dark in the way there is for us. There is only the world as it is cold, dark, pressurized, full of chemistry and biology, and the light that its own body produces, and the responses of other organisms to that light. The rest is silence. Not the silence of absence, not the silence of a world that has fallen quiet after having been loud. The silence of a world that has never been loud, that operates at frequencies and intensities so different from the world we know, that our words for sound and silence, light and dark, do not quite fit it. When you close your eyes to sleep, and the darkness of your room becomes the darkness behind your eyelids, you are moving towards something that the deep ocean has always been. A state in which the external world is suspended, in which the internal productions of your nervous system are the primary reality, in which the meaning of what you perceive is generated internally rather than received from outside. The deep ocean exists in a permanent version of this state. The organisms in it have evolved within it. They are, in the truest sense, creatures of the dark. Not in the way the phrase is usually used, as a metaphor for things that are hidden or dangerous, but literally. Organisms for whom darkness is not a problem to be solved, but a medium to be inhabited. They have made the dark their home, and they have done it by learning to make their own light. The blue glow of the anglerfish's lure, the brief flash of a dinoflagellate disturbed by a passing current, the long, slow pulse of a siphonophore drifting through the midnight zone, the private red spotlight of the dragonfish, invisible to everything around it except its prey. These are the lights of the deep ocean. They have been here for 400 million years. They are here right now. In the cold and the pressure and the silence and the dark, something is glowing, not for us. But if you are still enough and quiet enough, and willing to let the darkness that surrounds you be what it is rather than what you are afraid of, if you can rest in the not knowing, the way the deep ocean rests in the not seeing, then perhaps you can hear in the silence between your own heartbeats the faintest echo of a world that has always been lit in its own way by creatures that found the dark and decided to glow. Sleep now. The ocean is still here. It always will be. We began this journey with a number, 76 to 80%, the fraction of deep-sea creatures that produce their own light. It is a large number. It is, in the context of everything we have covered, a number that deserves to be revisited. The deep ocean is the largest living space on Earth. In that space, most things glow, not incidentally, not rarely, not as an exotic exception to the general darkness, but as the standard, as the norm, as the thing that the majority of organisms in the majority of this planet's habitable volume do as a matter of course, as part of the basic project of existing. We live on a planet where light is produced not just by the star at the center of the solar system, but by the organisms that inhabit it. We live on a planet that glows from within, not metaphorically, but literally. In the cold and the dark and the pressure of the deep ocean, where billions of organisms are producing photons right now from the chemistry of their own bodies. This is not a small fact. It is one of the central facts about the kind of place this is. The Earth is not a rock illuminated by a distant star. It is a living world that produces its own light through organisms that have been doing so for longer than anything on land has existed. The study of bioluminescence is, in this sense, the study of something fundamental about what life is and what life does. Life does not merely respond to light. Life produces light. Life has been producing light in the dark for 400 million years, and it will continue to produce light in the dark long after every human institution and every human memory and every trace of human existence has been dissolved back into the chemistry from which it came. The ocean will still be here. Its creatures will still be glowing. The anglerfish's lure will still be burning in the dark, patient and persistent and utterly indifferent to whether anyone is watching. And in that patient, persistent, unwatched glow, there is something that feels, if you let yourself feel it, like the most ancient form of reassurance that exists. Not that everything will be all right, not that anything is watching over you, but that the world is very old and very large and very full of things that have been doing what they do for a very long time, and that your brief presence in it, your brief, luminous, wandering presence in it, is part of something that was here before you arrived and will be here after you are gone. Rest now. The deep ocean is doing what it has always done. It is glowing in the dark for reasons of its own, and it is beautiful. There's one more thing worth saying before this journey ends. The photons produced by bioluminescent organisms do not disappear. They travel at the speed of light, the same speed at which the light from distant stars travels across the universe. They move through the water, absorbed by molecules, scattered by particles, converted to heat, or in rare cases, detected by the eye of another organism and transformed into a neural signal that carries information into the nervous system of something that was waiting in the dark. Most bioluminescent photons travel only a few meters before being absorbed. The ocean is not transparent. It converts light to heat with considerable efficiency. The light of a single dinoflagellate flash travels perhaps 2 or 3 meters before it is gone. The light of an anglerfish's lure travels perhaps 10 or 20 meters in the clearest deep water. The signal range of bioluminescence is local, measured in meters, not kilometers. But the chemistry that produces those photons has traveled further. It has moved through time. The luciferices and luciferins that produce bioluminescent light today are descended from molecules that produced light 400 million years ago. Not the same molecules chemistry degrades and is replaced, but the same lineage of chemistry, the same evolved solution to the same ancient problem, passed down through generations of organisms too numerous to count, refined by selection at every step, still operating today in a form that is related to its ancient ancestor in the way that you are related to yours. In this sense, every bioluminescent event in the deep ocean tonight is connected to every bioluminescent event that has ever occurred in the deep ocean. Not by direct continuity, the molecules are different, the organisms are different, but by the continuity of evolutionary descent. The light produced tonight is the light of a lineage. It carries, in its chemistry, the record of 400 million years of survival. This is what it means for something to be alive. Not just that it exists and functions in the present moment, but that it is the current expression of a process that has been running for a very long time. Every organism is a solution, an imperfect, ongoing, continuously revised solution to the problem of existing in a particular environment. The bioluminescent organisms of the deep ocean are solutions to the problem of existing in the dark. They have been refined by the deepest ocean on the slowest timescale, and they work. They glow. In the cold and the pressure and the dark, they glow. And that glow is not just light. It is history. It is the accumulated result of everything that worked and the absence of everything that didn't, over a span of time that makes the entire history of human civilization look like a single afternoon. The ocean is patient. Life is patient. The dark is patient. And the light, when it comes, is brief and cold and sufficient, exactly what it needs to be, in exactly the right wavelength, for exactly the purpose it has been refined to serve. This is enough. This has always been enough. Sleep well.